we're going to be talking about genetics and inheritance. Here we're going to explore how traits are passed from one generation to the next. Every one of us carries a genetic code that tells us how our cells function, how we look, and even how we respond to the environment. Think of this as our biological storybook written in our DNA. Genetics is the study of how traits are transmitted through generations, and it all starts with DNA, or deoxyribonucleic acid. DNA carries instructions for building proteins, which performs nearly every job in our cell. When we talk about the genome, we're referring to the sum total of all DNA in an organism. Humans inherit two sets of DNA, one from our mother and one from our father. Because of how genes get shuffled during meiosis, we're a unique combination of our parents' genetic materials. Even siblings differ because their process of genetic recombination ensures each gamete carries a distinct genetic mix. Our genetic information is packed into structures called chromosomes located in the nucleus. Each chromosome is a single continuous molecule of DNA wrapped around proteins for organization. When cells prepare to divide, these chromosomes become visible and duplicate into identical sister chromatids joined at a centromere. Each chromatid carries the same genes, which are specific regions along the chromosome that code traits. During cell division, these chromatids ensure that the proper transmission of genetic material to the new cell. Humans have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes that determine biological sex. The autosomes carry genes for most of our body traits, while sex chromosomes, the X and the Y, carry genes involved in sex determination in some unrelated traits. The pair of autosomes are called homologous chromosomes, meaning they are similar in size, shape, and gene arrangement. Each pair represents one chromosome from each parent, forming the foundation for genetic inheritance. Homologous chromosomes look alike but are not identical. Each carries the same genes, but those genes might exist in different forms called alleles. For example, one allele might code for brown eyes while another codes for blue eyes. When both alleles are the same, we call the person homozygous. When they differ, they're called heterozygous. Alleles arise from mutations, some changes in DNA sequence that introduce genetic variation. This variation is the raw materials for evolution and is why we see so much diversity within our species. A karyotype is a visual representation of the chromosomes in a cell, arranged from largest to smallest. It's a powerful diagnostic tool that allows scientists and doctors to identify abnormalities in chromosomes under the structures. When you look at a karyotype, notice how each pair aligns except for those sex chromosomes, which differ in appearance. The X chromosome is large, containing many genes, while the Y chromosome is much smaller. Karyotyping helps identify conditions like Down syndrome or Turner syndrome before or after birth. Your genotype is the complete set of all alleles, the specific genetic instructions you carry. Your phenotype, on the other hand, is what those genes actually produce, your observable traits. Hair color, eye color, height, even your risk of certain diseases are all influenced by your genotype. However, environment and lifestyle play a big role too. For example, a person may inherit the gene for tall stature, but poor nutrition during childhood can prevent that potential from being reached. So genotypes provide the blueprint, but environment and experience shapes the final result. To predict how traits pass from one parent to the child, we use a Punnett square, a simple yet brilliant genetic tool. Each parent contributes one allele for a given gene. By placing the possible alleles for each parent on the grid, we can visualize all potential combinations for the offspring. These combinations help predict probabilities of inheriting certain traits. It's important to remember that Punnett squares show possibilities, not guarantees, just like flipping a coin. Genetics involves chance within predictable limits. Gregor Mendel, which was an Austrian monk, laid the foundation for modern genetics in the 1850s using the pea plant. He noticed consistent patterns when breeding plants with contrasting traits like tall versus short or green versus yellow peas. From these results, he formulated two key laws, the law of segregation and the law of independent assortment. The law of segregation states that the allele pairs separate during gamete formation, meaning each gamete receives only one allele per gene. The law of independent assortment tells us that the genes for different traits are inherited independently of one another, unless they're linked to some chromosome. Not all alleles have equal power. A dominant allele masks or suppresses the expression of a recessive one. If you inherit a dominant allele, that trait will show up even if the allele is recessive. Recessive alleles only appear when both copies are received. Think of brown eyes and blue eyes. Brown is dominant, so even one copy of brown allele will override the blue one. Let's look at a freckles and ability to taste PTC, a chemical that tastes bitter to some people. If you have at least one dominant allele for freckles, you'll have them. If you have two recessive alleles, you won't. For PTC tasting, having one dominant allele means you can taste the bitterness, while two recessive alleles means you can't. These examples show that genetic expression is binary for some traits. You either show it or you don't, even though many traits are more complex. Sometimes recessive alleles carry harmful consequences. Take cystic fibrosis, which is caused by the mutation in the CFTR gene. People with two defective alleles lack the proper chloride transport protein, leading to thick mucus that clogs the lungs and digestive tract. Those with one normal and one defective allele, also known as carriers, don't show symptoms because the normal allele compensates. This pattern shows how recessive diseases can persist silently in a population, passing through the carriers who remain unaffected but still can transmit the allele to their children. In a common misconception, the dominant does not always mean common or strong. Dominance simply refers to how one allele masks another in heterozygous pairs. Some dominant alleles cause diseases such as polydactyly, or extra fingers or toes, or achondroplasia, which is a form of dwarfism. In those cases, having just one copy of the dominant allele is enough to produce the trait. So dominant doesn't mean normal or frequent. It describes how alleles interact, not how widespread they are. When studying two traits at once, we can use multiple complex Punnett squares to explore how alleles sort independently. For example, if one parent is heterozygous for both freckles and tasting ability, and the other lacks both traits, their offspring will be heterozygous for both genes. When two heterozygous males we get a variety of combinations. This demonstrates the law of independent assortment, where genes on different chromosomes are distributed independently, creating immense genetic diversity in the offspring. Not all traits follow simple dominance. In incomplete dominance, heterozygotes show a blend of both alleles. Think of hair texture. Straight hair comes from one allele, curly from another, and wavy hair results from having one of each. Another example is familial hypercholesterolemia, a condition affecting cholesterol levels. People with one defective allele have twice the normal cholesterol levels, while those with two defective alleles have extremely high levels of serious heart problems. This pattern shows how some traits display a gradient rather than an all or none expression. Codominance is a fascinating twist of inheritance. Instead of one allele overshadowing the other, both are expressed equally in the heterozygote. The ABO blood group is a perfect example. The A and B alleles are both dominant, so if a person inherits one of each, they express both types A and B blood. This means that their red blood cells carry both A and B antigens. Codominance shows how complex genetic expression can be, breaking away from a simplicity of classic dominant recessive patterns. The sickle cell gene is a real-world example of codominance. Here, there are two different alleles for hemoglobin. Hemoglobin A for normal hemoglobin and hemoglobin S for the sickle form. People who inherit two HBS alleles develop sickle cell anemia, a serious blood disease where red blood cells become crescent shaped and less efficient at carrying oxygen. These cells can block the flow, leading to pain, fatigue, and even organ damage. However, individuals with one HbA and one HbS allele, the carriers, produce both types of hemoglobin. They're usually healthy but may experience mild symptoms under low oxygen conditions. Sickle cell disease primarily affects people of African and Mediterranean descent. In the United States, about 1 in every 365 African American is affected. What's fascinating is how this harmful mutation persists in certain populations. It provides a survival advantage against malaria. People who are heterozygote for the sickle cell allele are less likely to die from malaria because of parasites can't thrive in their slightly altered red blood cells. This is a striking example of natural selection maintaining a trait that's harmful in one context but protective in another. Many human traits aren't determined by a single gene, but by multiple genes working together. A pattern called polygenic inheritance, where traits like height, eye color, and skin color and polygenic. Instead of discrete categories, these traits show continuous variation, forming a spectrum across the population. That's why height ranges smoothly from short to tall, and why skin tone varies gradually rather than over a fixed step. The more genes involved, the more possible combinations, producing an almost infinite diversity of human characteristics. Our genes don't work in isolation. The environment has profound influence on how they're expressed. Nutrition, exposure to sunlight, toxins, and even stress. All of these factors can modify gene activity. For example, identical twins share the same DNA but can look and behave differently as they age because their environment shapes gene expression in unique ways. Genes provide the blueprint, but environment determines how much of that blueprint is built, remodeled, or even silenced over time. Epigenetic inheritance takes this idea further. It refers to the changes in gene activities that don't involve altering the DNA sequence itself. Environmental factors can attach chemical tags such as methyl groups to DNA or modify these tightly in wraps around histone proteins. These changes control how accessible genes are for transcription, influencing when and where certain genes are turned on or off. Over time, these patterns can be passed to daughter cells and, in some cases, even to the next generation. Epigenetics helps explain why identical twins become more different over time, despite having identical genomes. Genes located closely to one another are the same chromosomes are called linked alleles. Because they're physically near each other, they tend to be inherited together. However, during meiosis, crossing over can separate them into two if it occurs between the genes. The farther apart two genes are from the chromosome, the more likely crossing over will occur between them. Linkage mapping, or tracking how often a certain trait are inherited together, has helped scientists identify the location of thousands of genes related to diseases in development. The 23rd pair of chromosomes determines biological sex and carries unique sets of genomes. The X chromosome is large and contains many genes unrelated to sex, such as those for color vision and blood clotting. The Y chromosome, much smaller, mainly carries genes for male determination. Because males have one X and one Y chromosome, any defective allele on the X has no counterpart to mask it. Females with two X chromosomes can be carriers without showing symptoms. Importantly, the father determines the sex of the offspring by contributing either the X or the Y. When genes are located on the X or the Y chromosomes, their inheritance follows sex-linked patterns. For X-linked recessive traits, males need only one copy of the defective allele to express that trait, while females actually need the two. This is why conditions like color blindness and hemophilia appear more often in males. Mothers can pass the X-linked trait to their sons, but fathers cannot, since sons inherit the father's Y chromosome. Daughters, however, can inherit the recessive allele from either parent and may become carriers even if they don't express the trait themselves. Let's look at hemophilia, which is a classic example of X-linked recessive disorders. People with hemophilia lack a functional blood clotting protein, causing prolonged bleeding after even minor injuries. A female with two normal alleles has normal blood clotting, while a carrier female has one defective allele but still produces enough of the protein to prevent symptoms. A male with a defective allele, however, will have hemophilia because his single X chromosome carries no backup copy. The inheritance patterns highlights how the same genetic conditions can affect men and women very differently based solely on chromosomal makeup. Sex-influenced traits behave a little differently because their expressions depend on the presence of sex hormones like estrogen and testosterone. The genes for these traits are located on autosomes, not sex chromosomes, but hormones affect whether their traits appear. Baldness is a great example. Both men and women can inherit the allele for their hair loss, but the expression is stronger in men because of higher testosterone levels. This is why baldness can skip women in families, yet show up predominantly in male relatives. Hormones can act like genetic light switches, turning certain traits on or off depending on their biological context. Sometimes errors occur during meiosis, the process that forms gametes. If homologous chromosomes or sister chromatids fail to separate properly, the resulting gametes may have too many or too few chromosomes, a mistake known as non-disjunction. When a gamete like this fuses with the normal one during fertilization, the zygote ends up with an abnormal number of chromosomes. This can cause conditions like trisomy, where one of the three copies of the chromosome, or monosomy, where there's one missing. These errors remind us how delicate and precise the process of genetic diversity really is. Down syndrome, or trisomy 21, occurs when an individual inherits three copies of chromosome 21 instead of the usual two. The extra genetic material affects development, leading to characteristic physical features and varying degrees of intellectual disability. It occurs in about 1 in every 800 live births in the United States. The likelihood increases with maternal age as the risk of non-disjunction rises in older eggs. Prenatal screenings can detect trisomies early, giving families time to prepare for the specific needs for the child. Despite the chromosomal differences, many individuals with Down syndrome live healthy lives, fulfilling lives, and proper support. Non-disjunction can also affect sex chromosomes, leading to several distinct syndromes. For example, XYY males, sometimes called Jacob's syndrome, tend to be tall but otherwise typical in development. Klinefelter syndrome, XXY, affects males who are usually tall but sterile and may have mild learning challenges or breast development due to the extra X chromosome. On the other hand, Turner syndrome, or XO, occurs in females who are only one X chromosome. They are usually shorter, sterile, and have subtle physical differences, though intelligence is typically normal. These conditions highlight how variations in chromosome numbers can alter development without necessarily affecting viability. Beyond changes in the number of chromosomes, also experience structural alterations. In a deletion, a piece of the chromosome breaks off and is lost. For example, the Creutzfeldt syndrome, typically cry of the cat, results from the deletion of chromosome 5 and causes developmental delays and distinct high-pitched cries in infants. In a translocation, a segment of one chromosome attaches to a non-homologous chromosome. This can disrupt gene regulation and increase the risk of certain cancers, such as leukemia. Structural mutations like these remind us that the genome isn't like a list of genes. It's an intricate architecture where small rearrangements can have big effects. Many genetic disorders arise when persons inherit two defective copies of recessive alleles, one from each parent. Carriers are typically healthy because their normal alleles compensate. And phenylketonuria, or PKU, is one such condition where individuals lack the enzymes to process phenylalanine, an amino acid found in many proteins. Without the treatment, it can actually lead to brain damage, but careful diet control can prevent symptoms. Another example is Tay-Sachs disease, which results from a missing enzyme that breaks down lipids in the brain. It's particularly common among the individuals of the Ashkenazi Jew descent and leads to severe neurological decline in early life. Huntington's disease is an example of a dominant lethal condition, meaning that inheriting even one copy of the defective allele leads to disease. However, symptoms don't appear until midlife, after most affected individuals have already had children and passed on the gene. The disorder causes progressive breakdown of nerve cells in the brain, leading to movement problems, mood changes, and cognitive decline. Its persistence in human population shows how genetics can defy usually natural selection when symptoms emerge after reproductive years. We've covered the vast amounts of information, but it all connects to one theme: variation. DNA carries the instructions for life, but the way those instructions are shuffled, expressed, and modified creates individuality. From Mendel's P-plans to modern genomics, we've learned that inheritance follows predictable patterns, yet it's full of exceptions like incomplete dominance, codominance, and polygenic traits. Mutation, chromosomes, errors, and environmental influences further shape our genetic identity. The complexity of inheritance reflects on the beauty of biological diversity and constant interplay between chance and order.